So, good afternoon. My name is Ling Ling. So I'm a social professor in Department of Biomedicine in Aarhus University, and also Standard Diabetes Center Aarhus, which is SDCA. And today I would like to give introduction about our platform, which we call SCOMICS, on single cell and spatial transcriptomics in AU and SDCA. So back in 2018, the NGS next generation sequencing, it was selected as the method of the year because it really scaled up the studies from gene by gene to the genome-wide scale. And 5 years afterwards, in 2013, single-cell sequencing was selected as the method of the year in Nature Methods because it increased the resolution of the studies. So there are a lot of good reasons why we focus on single-cell RNA sequencing. So it's basically what we can do with single-cell RNA sequencing. So if we think about the RNA sequencing as we are taking a look at the gene expression profile at genome-wide, but at average value of different genes from the same tissue, and the single-cell RNA sequencing allow us to take at the genome-wide transcriptome on each of the single cells, which allow us to identify the heterogeneity within one tissue. And in this case, we can identify, for example, the novel cell types, and also we can compare between different groups. So we could if we compare the disease group with the control group, we can identify some of the disease-specific populations and also what the patterns on the gene expression for the disease groups. So there could be some like potential target for the therapeutic strategies or some early biomarkers. So there are a lot of advantages to use the single-cell RNA sequencing. And also, we can work on the single cell studies in different dimensions if we take a look at the different part of the centroderma. So we can work from DNA to RNA to protein, so which means genome, epigenome, transcriptome, and protein, or the combination of some of them for the same single cell. And all these years, a lot of methodologies has been developed to improve the scales about how many cells that we could include it in one project, and also how many samples that they could involve in one project. And you can see now the numbers is dramatically increased. And another method of the year in 2020, and that is the spatial technology. It is not an alternative way of single-cell RNA sequencing, but it's like a complementary to single-cell RNA sequencing, because single-cell RNA sequencing gives us very good resolutions to look at the gene expression. But while we're isolating the cells, we lose the original spatial information. So the spatial sequencing allows us to take a look at the genome-wide transcriptome patterns on the same sections at the same time. So here is how our journey started within this field. Because we see this big potential of the single-cell RNA sequencing, in 2019, a few groups in Aarhus, we have collected some budget to buy this little guy, 10x Chrome controller, and establish the single-cell RNA sequencing platform in Aarhus University. And right now, our single-cell RNA sequencing lab is located on the first floor in Aarhus campus school building. And this lab is next to the FACS core, so it makes life much easier if we would like to enrich some of the cells that with interest. So from 2019, and with this platform, we have worked with different kind of libraries for single cell. For example, the 3' libraries, 5' libraries, VDJ, feature barcode, and also the CellX. And in 2020, we have also established the single-cell RNA sequencing platform with the BD Rhapsody systems on the title panel and also the VDJ libraries. In 2021, we established our platform on spatial sequencing with 10x Genomics visual systems. At the same year, we also established the platform to convert the single cell or spatial barcode amplified full-length cDNA into the nanopore library, so which allow us to take a look at the isoform preference at single cell resolutions. In 2022, in order to move the translation medicine forward, we set up our new home in FDCA, which is located on the 4th floor in Aarhus University Hospital in Skadi. So in our new lab, we have these two members. One of these upgraded Chrome controller from X, which allow us to do the high throughput single cell library generation. And also we have the site assist, which allow us to work with the FPE fixed samples. And on the same floor, we have the section lab, where we have the cryo state, so we can prepare the samples for the spatial sequencing. And also we're going to have the FACS core with one analyzer and one sorter, so which we could pre-enrich some of the cell populations before the single cell studies. And this year, in 2023, we have planned something. So the first is we are planning to establish the high resolution spatial sequencing platform with stereomic system from MGI, which gives the resolution to nanoscale. And also with this platform, we're quite flexible on the sample size. So we can work on the sample from millimeter scales to centimeter scales. We also have the sequencer in our lab from MGI and DNB6 G400, which allow us to do a quick sequencing after we generate our library. Also, we're planning to develop the automation systems on library generation, so which will save us more time to have more fun on the data mining. Beside of the wet lab platform, we also developed the pipelines for the data analysis for spatial sequencing, single cell RNA sequencing, and also the isoform preference analysis. So I'm not going to go through all these details on the pipelines because that would be really time consuming. So I will, we can talk about it later in some other seminars. I know coding is a little bit tricky for some of the labs. So we also developed the server for the single cell and spatial sequencing data visualization and sharing. So once the basic analysis were performed, we can put the single cell or spatial data set into our server in Aarhus University. So in this case, you can just simply type in the name of the gene. So you can visualize the gene expression patterns in different ways. So UMAP, TC, violin plot for visualization. And you can visualize the expression of multiple genes with dot plot or the heat map. And also you can select in which way that you would like to group your cells, if by sample or by different cell types or by different groups. And this server is based in Aarhus University, but you can access it from anywhere in the world from your laptop, and it's quite easy to work with. Just simply type in and click, and you will get all the plot that you would like to get. For now, we have more than 10 projects that we have in our server already. So all these years, we have over 30 projects in collaboration with different groups, and we have worked on different species and different organs and different libraries, and also end up with different publications.